Thank you everybody for being here with me today. I will try to make you understand what I've done for the last 6 years and what are the important results from my work. This is Björk. She's a great singer, and she comes from Iceland, which is an oceanic island with volcano, just like São Tomé and Príncipe. And she's wearing a dress representing an orchid avatar by Iris van Echten. And Iris van Echten is a fashion designer, Dutch fashion designer, who thinks that fashion could be bringing closer to nature, science, and biology. And her last collection is inspired by a book on fungi written by Mardulyn, this guy here. And Mardulyn is a tropical ecologist, but he's also the son of another biologist, the biochemist Rupert Sheldrake. And so Rupert passed to his son, the next generation, the characteristic of having an interest for science and loving biology. And so Mardulyn takes that characteristic a little bit differently from his father, from biochemistry to tropical ecology, and that's in a sense what evolution is, so the passing of character from one generation to the next with slight changes, mutation, and evolution was one of the big areas of research for my work. And well, Iris van Echten also brings couture and biology together by designing the Naturalis Biodiversity Center in Leiden, which is the Natural Museum of the Netherlands. And just like our museum in Belgium, that contains millions of biological objects. And my favorite objects are the herbarium specimen. So it's plants that have been drying and bring back from all over the world by scientists, botanists for the last 2 centuries. And this is to preserve them and to have a knowledge on the diversity in the world. And that brings another area of research for me, taxonomy. So when we speak of biology, taxonomy, and evolution, there is generally 2 people that are mostly cited, which is Charles Darwin and Linnaeus. And here too, there is some historical familial history, because the grandfather of Charles Darwin translates in English the Species Plantarum of Linnaeus, so the big book he writes. And so Charles Darwin gets his idea on evolution by doing a big journey on a boat, the HMS Beagle. And this is an image taken from a movie made on that trip, a movie made a couple of years ago. That's a really good movie on his life and his travel. And on the other side, Linnaeus, he's a Swedish naturalist and is considered as the father of modern taxonomy, with the genus name associated with a specific epithet. So if you bring like the cat, it's Felis catus. Felis is the genera, the genus name, and catus the specific epithet. And one of the most famous quotes of Linnaeus is that if you don't know the names of things, the knowledge on them is lost too. And I think it's really true and really important, and that some of the results of my thesis are very in line with this quote. So my thesis was at the intersection of several disciplines of biology. So there is taxonomy, and in particular on the Orchidaceae. And then I did phylogeny to then obtain a biogeographical history of the genus. And then at the species level, so population level, I did some phylogeography. And now I will try to make you understand a little bit more what it is, the generalities on this different concept. So, Orchidaceae, why did I choose this family? So, first of all, because of their beauty and diversity. So, yeah, orchids are most probably the biggest, largest family of flowering plant in the world, with 28,000 species. And they are found mostly on the tropics, but also in temperate region, so from one polar circle to the other. And
So yeah, on this slide you can see that they have just an amazing variety of shapes, and there is some orchids with flowers in the shape of a duck, or a dancing fairy, of a bird, this one is upside down, or the face of a monkey, and well, this one is called the naked hangman, and I guess you can see why. And talking about inappropriate and very sexually related plants, well, orchids, the names come from the shape of the roots of some European orchids that are tubers, able to store resource, and that looks like orchis in Latin, which means testicles. So yeah, that's a first link. And then also another link between sexuality and orchids is that a lot of their diversification worldwide is linked to interactions with pollinators. So you know that the flower are the sexual organs of plants. Yeah, so orchids are hermaphrodites. They have both male and female organs on one flower. And it's better to make the female part of one flower, of one plant, meeting the male part of another in order to mix genes and avoid inbreeding. And that's the tricky part when you don't move. So some plants are using the wind to do that, and especially orchids, are using pollinators, and a lot of a massive range of interaction with pollinators. So if you take by example this Ophrys apifera, which is an orchid that was found not so long ago on the other campus. So don't you think the labellum, this petal, looked just like a nice female bee? Well, the male bee believes that too. So when it's the mating season, he will approach the flower, he will go onto the flower, and by doing that, he will attach the pollinia, so the two masses of pollen there, onto his head. It would stick onto his head, and then, well, after a while, he will realize, oh, it wasn't a female bee, but he will see another one just next to it, and he will go on that one, and that's how he brings the pollinia from the first flower to the female part of the other flower, the gynostemium. And as you can see here, this poor guy, he tried a lot of time and it was not really successful. And well, from the sexuality of orchids to Darwin, there is also a link, yeah, believe me, because he wrote one of his books, one of the most famous books on orchids, and he wasn't interested in the sexually deceptive orchids like this one. No, he was interested in the one that gave something to the pollinator, the rewarding one. Like this Angrecum that is found in Madagascar. And he predicted that if there was a flower with a spur, so this organ that stores nectar, a spur that is 40 centimeters long, there should probably be a moth that have a proboscis that is 40 centimeters long too. And that pollinator have been found 50 years later. So, again with Darwin, because I finished with Darwin, we go back to Darwin and the first word of my title, which is biogeography. So when he embarked on his trip on the Beagle, Darwin was interested in the natural history of the different regions of the world. And the trip lasted for 5 years, and he went from Europe around South America, and then up to New Zealand, Australia, Asia, and then around Africa. But one really important part of this trip was the Galapagos Island. Again, an oceanic archipelago. And he said that in his second chapter of The Origin of Species, that it was this trip and this particular moment that helped him to get ideas for his theory on evolution. And so at that time, he said, many years ago, when comparing and seeing that others compare the birds from the separate island of the Galapagos archipelago, both one with another and with those from the American mainland, I was much struck how entirely vague and arbitrary the distinction between species and varieties. And then, seeing this gradation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds in the archipelago, one species has been taken and modified for different ends. And so the theory on evolution that was written and conceived by Darwin, but also Wallace, the guy there, is really an approach that has been taken by a historical approach on the geographical distribution of organisms, and that's exactly what biogeography is. And Wallace is considered to be the father of biogeography, and it's really their collaboration and a long correspondence between the two guys that helped them create their theories and thinking about it. So yeah, that was something really important. And then for phylogeography, so phylogeography is the study of the formation of species, so at the population level or at the individual level. And I thought that maybe a way to make you understand what phylogeography or biogeography is to take an example that everybody has, so the family. And in this case, let's say we are aliens that are going to look at the biogeographical, phylogeographical study of this part of my family. So there is two people from Poland here that have blonde to brown hair, light brown hair. One guy in Belgium that have brown light hair. Two people in Belgium with brown hair and a lot of whites. And then two redheads, one in Belgium and one in the south of France. So when you look at their morphology, you will think, oh, we can group them by the color of the hair. And because there is more people in Belgium, you can assume that Belgium is the center of origin of this group of organisms, and that there was two events of colonization, one to Poland and then one to the south of France. But then if we look at the genetic markers and at the genealogy, the story is a little bit different. And yes, the fact that there are, so this is me, my brother, our closest cousin. On our grandfather's side, one girl, and on our grandmother's side, the two persons in Poland and one person in France. And so the fact that we are more numerous in Belgium is only due to the fact that there was two events of dispersion from Poland to Belgium, and the fact that there is this girl in France is a long distance dispersal from Poland to France. And so that's, in a way, what I applied to my orchids in order to retrieve a more to retrieve a better tree of species, and also the relationship between individuals for one species of orchids. So from the title, I really have to go through one last part, which is the area of study, and it's São Tomé and Príncipe. So São Tomé and Príncipe are two islands in the Gulf of Guinea, and they are part of the Cameroon volcanic line, which go from the Lake Chad up there up to Annobon. And São Tomé and Príncipe are part of the Gulf of Guinea archipelago, which is composed of 4 islands. One continental island, Bioko, that have been connected to the continent multiple times through time, and then Príncipe, São Tomé, and Annobon, that are oceanic islands born from the volcanism in the region. So São Tomé and Príncipe have been discovered during the 15th century by the Portuguese, and there was no human on this land, only birds. And so they decided that it would be a nice place to start a laboratory on the slave trade and on the plantation of coffee, cocoa, and sugarcane. And that gives to São Tomé her name of Chocolate Island. So Príncipe is the older of the oceanic island. It's 31 million years old, and it's the closest to Equatorial Guinea. And in 2012, it has been included in the Man and Biosphere Reserve of the UNESCO. São Tomé is much more younger, 13 to 15 million years old, and it's a little bit further from the continental coast, 239 kilometers from Gabon. And São Tomé is much more larger and higher than Príncipe too. Príncipe is up to 950 meters altitude, and São Tomé is up to more than 2,000 meters altitude. So São Tomé and Príncipe are mostly known for their endemic birds. So endemic means that they are found nowhere else than on these islands. And so they have a very large number of endemic species. So 17 species for São Tomé, 8 species for Príncipe, and 3 species shared by both islands. And in comparison, the Galapagos, they have 22 species of birds. But if you take all the Galapagos Islands together, it's 10 times larger than São Tomé. So it's a really big number compared to the size of the 2 islands. And here you have the smallest ibis in the world and the biggest canary. And they are both critically endangered on the IUCN Red List. So the IUCN Red List is an indicator of the health of the world's biodiversity. And it's used by multiple actors to inform and address questions on biodiversity and take decisions for conservation. And critically endangered is the last step before extinct. So it's some species that are really to protect. And concerning the endemicity of the flora of the islands, it's quite high, an endemic rate of 12.5%. It's quite high. And there is 3 families that account for 50% of this endemic species. It's the Orchidaceae, the Rubiaceae, and the Euphorbiaceae. And here it's Polystachya, an orchid, and Psychotria, a Rubiaceae, and it's the 2 genera with the most number of endemic species for the archipelago. Here you have the giant begonia, so also endemic from São Tomé and Príncipe, and the leaves are that big, so it's quite impressive. And 54% of the species found in the archipelago are endemic. And for the impatiens that are here, it's the 3 species that are endemic, 3 species that are found in the archipelago that are endemic, so 100% of endemicity. So concerning, of course, the Orchidaceae, it's 101 taxa, so species and subspecies on São Tomé, and 71 on Príncipe. And 75% of them are epiphyte. That means they are growing onto the tree, on the bark of the tree, on the trunk, or on the branches. But that does not mean they are parasites. They do not harm the tree. It's just that they use it as a support, and they use their roots to take nutrients and water from the atmosphere. So they are mostly found at higher altitudes, where they are in the cloud forest, where it rains a lot, and where the humidity in the atmosphere is higher than anywhere else. And so here you have 2 species of Tridactyle, so my genus of interest. And it's called Tridactyle because you see the labellum, it's divided into 3, and especially for this species, it really looks like 3 finger, which in Latin is tridactylis, so Tridactyle. And now you can ask, but she told us that Polystachya was the most numerous genera with a lot of endemicity. This archipelago. Why Tridactyle? Well, that comes from 2 questions, and the first one was a taxonomical question raised by 2 different studies. So as biologists or systematicians, we like monophily. So we like when all the species of one genus are grouped and closer together than to any other species of any other genus. And well, here you have Tridactyle, Ypsilopus, Rangeris, Tridactyle, Ypsilopus, Podangis, Rangeris. So it's just a mixed match, and that's kind of problematic. So that was the first question raised by those two studies. And then another question was raised by two master theses done at ULB. The first one by Sophie Le Guil that worked on the phylogeny of Tridactyl, and then the second, well, done by me on the taxonomy of Tridactyl-Tridactylites, which is one species that wasn't monophyletic. So here with the rectangle and solid line, you can see, well, it's all specimens from Tridactyl-Tridactylites. So that was another question, and especially the fact that in the green lines here, all the specimens are coming from São Tomé. So that leads to the goal of research of this thesis, which was to better understand the mechanism of diversification of the African flora in the Gulf of Guinea island. And I focus on Tridactyl to unveil the scenario leading to a high endemism on these islands. So I had three objectives that are three chapters. So the first one was to reassess the monophyly of the genus and to evaluate the diversity of Tridactyl species on São Tomé and Príncipe. Then for the chapter 2, obtain a better result phylogeny for the genus. And then dating this phylogeny and doing a biogeographical analysis to assess the history of the genus. And then in last chapter, to study the geographical distribution of the genetic variation of that particular species, Tridactyl-Tridactylites, with approximate Bayesian computation analysis. I will go back on that later. So that's the most fun part of the thesis, which is going onto the field to collect, look at, and observe my species. And so here you can see that I'm on the fallen trees. And usually we do this kind of rescue operation, so we don't harvest the whole forest from all the orchids there. We're just taking them when they are on fallen trees and that they are going to die there. And if really, like here, it's some Tridactyl, if really I need a sample of plants on living tree, then we take 1 or 2 samples when there is at least 10 in the tree. And so, yeah, I spent 80 days on the field in Gabon, São Tomé and Príncipe. And when we collect these orchids, we don't bring them living here. We let them in the shade house network. So it's a project started 20 years ago by Jean-Philippe Biteau, and that have been used in multiple countries in Africa by Tariq, Stévart, and collaborators. And so when we found the plant on the field, we don't find them with flower. Usually they are non-flowering. So we bring them, we put them on planks, and then we hang them in an open area with the material on top to protect them from the sun. And then they will grow and they will continue living. Some of them are living in São Tomé since 20 years. And so this was a project that started 20 years ago, and that we have 30,000 living plants actually on site. And more than 60 new taxa, so genera, species, subspecies, have been described from that system, because when finally they flower into the orchid shade house, then we photograph them, we collect the herbarium specimen, the alcohol preserved specimen, and the DNA for phylogenetic analysis. And that system allowed me to collect 200 samples of Tridactyl for phylogenetic analysis. So for the molecular analysis, I used the PCR Sanger method with 5 plastid marker and 1 nuclear marker from the ribosome. And yeah, because of the pandemics, it's exactly the same technique as used by most medical laboratories all around the world. And I know that most newspapers have explained way better than me how the PCR works. So that allowed me to skip that part. And so the result with the phylogenetic analysis is this one. So with collaborators, we described 2 new genera, Aziza and Planetangis, and the genus Rangeris is now a synonym of Podangis. And this is a drawing that I've done of Aziza. And Aziza is a name that is inspired by a fairy from the Bantu culture, so in the Nigeria specifically, where Aziza is a fairy that hides with a very long hair, just one hair. She's hiding into that hair, and that makes her hide in the forest. And we took that name because of the very long spur of this Aziza trilobata. Concerning the monophily of Tridactyl, the genus Tridactyl, so the monophily was reassessed by transferring 5 species into the genus Ypsilopus. And that was done using phylogenetic tree, using the 6 markers that I previously said. And one particular morphological characteristic that is a very small characteristic, it's the stipe. So it's the part that is sticky and that holds the pollinia together onto the plant. And in Tridactyl, the stipe is in the form of a diamond, and in Ypsilopus it is bifid, so just like a Y-shaped, and that gives the name to the genus, because epsilon, it's Y in Greek. And so the second part of this chapter was to examine the morphological diversity of Tridactyl species. So I observed more than 1,000 herbarium specimens and living plants. And again, with collaborator, we described 2 new species from East Africa, one Tridactyl from the section Egelingii, that was a former genus, and then one Ypsilopus. And so here you can see the stipe that is wide-shaped. And finally, concerning the orchids of São Tomé and Príncipe, so we described 4 new species from São Tomé that are endemic of the archipelago. So Tridactyle-Elżbietae, that is dedicated to an orchidologist that died in 2017. Tridactyl-Levlevae, because lev is the word in São Tomé to say slowly, and it took us a while to understand the taxonomy of that species, so slowly. Nucifera, because it's the one I showed you before that was growing onto a coconut tree, and coconut tree, the Latin name, it's Cocos nucifera, so Tridactyl nucifera. And the last one, odorata, because from dawn and until late in the night, it smelled really good. So concerning the two following chapter of the thesis, the nuclear markers we get with Sanger PCR were not sufficiently powerful to retrieve the story we wanted, to the phylogeny we wanted. So we use another technique, the next generation sequencing Illumina, and one particular technique which is the genome skimming. The genome that is cut in very little pieces, the reads, and there is a lot of them, and they are slightly overlapping with each other, which allow us to reassemble after the genome. And with the genome skimming, so of course, if you have a lot of reads, you have a lot of information and you are more confident on the information at each position, but that costs a lot of money. So genome skimming is a way to avoid the fact that it costs that much money. So you sequence the whole genome at a very low coverage, but you are interested in only the part of the genome that are found in more numerous in the different cells, so the plastid genome or the nuclear gene that are coding for the ribosome. And so even if it's at low coverage, you still have a very high chance to have the whole chloroplast and the whole ribosomal region. So we did that, and that worked pretty well, and we were even able to do it on herbarium specimen and on type specimens, so the specimen used to describe the species. It's pretty cool, and that wasn't done before for the Sanger PCR method that did not work. So we were really happy with that result, and it allowed us to have 220 specimens from 61 taxa and 34 tridactyl species that were already described. And so after the assembly, so we did the phylogenetic, we retrieved the phylogenetic relationship. We estimate them with 3 different methods: maximum parsimony, maximum likelihood, and Bayesian inference. And then the phylogeny was dated with BEAST, the little octopus there, so Bayesian inference. And then for the biogeographical part, we use RASP, which is a reconstruction of ancestral states in phylogenies, to have the ancestral area of the phylogenetic tree. And then for the chapter 3 on interspecific level, we use DAY-ABC, which stands for do-it-yourself approximate Bayesian computation to retrieve a scenario of phylogeography for the species Tridactylites. So these were my work hypotheses for the second part, so the biogeography. And to understand how the endemic species were arrived in São Tomé and Príncipe. So the first one is colonization without speciation. So you see there is 2 flowers that came from the continent to the islands, and then the green one disappeared on the continent. So this is an hypothesis of paleo-endemism. That means that, at least our definition, is that the species was present on the continent and then disappear on the continent. And then second hypothesis is the adaptative radiation. So one species go from the continent to the island and then speciate there in multiple species with one single ancestor. And then the third hypothesis was colonization involving ecologically or taxonomically related form. So here it's more, so the species speciate on São Tomé and Príncipe or in the continent, and then there is exchange with possibly extinction. And well, it's more about neo-endemism for the archipelago, so that the species speciate on the island and not because they were extinct on the continent. So that were my 3 hypotheses. And this is the result I get for, so this is the phylogenetic tree obtained for the ribosomal combined with the chloroplastic data set. And these are the posterior probabilities obtained with the Bayesian inference, but the results were similar for maximum parsimony and maximum likelihood. And the results were also similar for the ribosome alone and the chloroplast alone, and they only differ with non-supported branch. So here you have the outgroups. In yellow, the Orchidoidae, which is another subfamily, and then the Epidendroidae in purple, and then within the Epidendroidae, you have the Vendée, which is the subtribe with the Tridactyl. And here you can see the different groups that were retrieved by all trees and all analyses, and these groups are characterized, each one, by morphological characteristics that are not retrieved for others. So, by example, the section Egelingiae contain all the species that were formerly Egelingia, and they have as characteristic the concave lip and no three-lobed labellum. And then here you have the group 4 with all the the species with fleshy leaves. So concerning the dating of the phylogeny, so this is the tree obtained when we date the phylogeny, and the blue bars indicate the interval of confidence for the nodes, the node age. So we can see that some are really big. We are not that certain of these nodes. So we calibrate the tree with 2 fossils of orchids. That's not a lot, but it's very difficult to obtain fossil for orchids. So that's what we've got. And then 2 calibration points that were retrieved from Givnish et al. For the separation between the Orchidoideae and Epidendroideae, and then for the Angreciinae, which is the group of Tridactyl. And, well, it's not very clear, but here you can see that the separation between the Tridactyl is dated more than 20 million years ago, so during the Miocene, just after the separation between the Oligocene and the Miocene. It's the period of the opening of the Red Sea, the surrection of the Atlas, the East African mountains, and possibly the surrection of the Cameroon margin and the late formation of the Cameroon volcanic line. And concerning the orchids of São Tomé and Príncipe, they are in color here, and you can see that they are distributed into the tree, and that their closest sister are from the continent and not from São Tomé or Príncipe. So in red, São Tomé, orange, Príncipe, and in yellow, it's the 2 that are found on São Tomé and on the continent. And so if they are distributed through the tree, the adaptative radiation hypothesis is not really possible, so that's the one we reject there. And then the dating of the phylogeny indicates that these species and their sister species are much more younger than the appearance of the islands. So it indicates more toward neo-endemism, so the formation of the species on the archipelago, than for paleo-endemism and extinction on the continent. So that's why we can tick the box for hypothesis 3, so multiple colonization and neo-endemism. So concerning the ancestral area, biogeographical analysis. So here you can see in teal, there is a lot of teal there. It's the Lower Guinea, so Cameroon, Central African Republic, Congo, Brazzaville, Gabon, Equatorial Guinea. And the most common ancestor of all Tridactyl have a probability of 100% to come from Lower Guinea. Concerning the species on the archipelago, it's less clear. It's usually multiple area probable, but the sister species of the species found in São Tomé and Príncipe are usually coming from Lower Guinea. So, comparing my results to the other phylogenies retrieved for plant on the archipelago, there is 2. One on the impatiens, and that wasn't dated, so they don't have the timing for this group. And all the species of São Tomé are grouped in one clade. So probably 1 or 2 events of dispersion, and this is more what you retrieved usually with adaptative radiation. So that's a potential explanation for this group. And this one has been dated, so it's the phylogeny on the begonia made by Plana in 2004. And here the history look more like the one I retrieved for Tridactyl. So it's during the Miocene for most of the species, and there is species on São Tomé and on the continent that are mixed in the tree. So more likely the same kind of story, maybe with paleo-endemism, so extinction on the continent, then apparition on the islands, but that's not that clear. So finally, that's the last chapter of my thesis, and that's on the geographical distribution of the genetic variation within one species. So I took a different specimen, different individual of that species, and I analyzed the difference between them genetically. And we did DAY-ABC analysis. So this is an analysis to retrieve a scenario of possible colonization. So I tried 4 different scenarios. The first 2 scenarios were a colonization from Príncipe to the continent, to the different area on the continent. So in black you have Guinea, in pink you have Cameroon, and then in yellow you have Gabon. And there is 2 scenarios because there is one recent scenario between the last glacial maximum and the penultimate last glacial maximum, so the one just before, and an ancient scenario, so before the penultimate last glacial maximum. And then the scenario 3 and 4, it's colonization from Cameroon to Príncipe and then to the other region on the continent, also one recent and one ancient scenario. And this is the result that we obtain with the DAY-ABC analysis. So each dot is one scenario with different parameters that were estimated by the program. And in yellow, you have the observed data. So the scenario 1 from Príncipe recent is in red. Scenario 2 from Príncipe ancient is in green. Cameroon recent is in pink, and Cameroon ancient is in blue. You can see that the observed data are just on the edge of the cloud of points for the scenario 1, so the back colonization from Príncipe to the continent. Well, the observed data are here on the edge, so it's not that clear, but that's the scenario we tested. That is the more probable with the data observed. And here you have another one with only from the continent to Príncipe. I test other scenario from the continent to Príncipe, and you can see that the observed data are very far away from the cloud of points. So that's just preliminary result. But anyway, we got them, and we also had the result that the population genetically, so they have a lot of genetic diversity, the same level as in Cameroon, which is much more bigger. You can see the different area we sampled, so much more bigger and less isolated that Príncipe is. So the fact that the genetic diversity is as high as in Cameroon is already interesting. So to conclude, so concerning the chapter 1, the reassessment of the monophyly of the genus. So we now have a genus that is monophyletic, and we describe 2 new genera, 5 new species of Tridactyl and 1 species of Ypsilopus. And I have a phylogeny that is pretty well sampled and resolved thanks to the NGS method. And it's in orange because we lack samples from East Africa, so maybe the history is a little bit different, but anyway, it will not change anything for the archipelago and the biogeography of the Gulf of Guinea island. So with the 4 species that we described, there is now 20% of the genus that is found in the archipelago, and the endemicity that was previously of 50% is now of 75%. So the archipelago is really sanctuary for the genus. And concerning the biogeography, it's most probable that there is multiple events of colonization from the continent to the archipelago, and that it's neo-endemism, so that the endemicity is from speciation on the island and not extinction on the continent. And then concerning the species Tridactyl-Trydactylites, so there is an important genetic diversity on Príncipe, just I said, and a potential back colonization from Príncipe to the continent, but that needs probably more sample in the different regions. So concerning the perspective for São Tomé and Príncipe flora, doing this kind of biogeographical studies on Rubiaceae, like this Psychotria, or Euphorbiaceae, like this tree, and on the Polystachya, which is the genera with the more endemic and the more species on the island. That would be great to see if the results are the same as I obtained or different. Concerning the orchids, so like I said, the sampling in East Africa was quite scarce, and also in Nigeria and Republic Democratic of Congo. So that would be nice to include that for my genus, but other genera of orchids from Central Africa in general. Nuclear markers. So I use chloroplastic markers and ribosomal gene, but there is the whole nucleus that contains other genetic markers, and that would be nice with new probes that have been developed recently, so the Angiosperm 353 project or the Orchidaceae 963 project, in order to see if there is discordance between the nuclear genome and the chloroplastic genome. And finally, concerning the pollinators and the fruit, so these are two visitors that I've seen on Tridactyl on São Tomé, one tipulid and one moth. I only said visitor because we are not certain yet that they pollinate the Tridactyl, so that's some study that have to be done for Tridactyl, but also for other angrecoid orchids and orchids in general on the archipelago. And then for fruit, the fruit of the orchids are important taxonomically. Their shape, the way they open with one valva or two valva, it's really important for taxonomy purpose and also for conservation. So looking more for the fruit for taxonomical, systematics and conservation purpose would be nice. And finally, at the global level, a big question, I think, is why some species, some genera radiate and other not. So this is a radiation on La Réunion of the Adrangis. And in my case, in the Tridactyl, they did not radiate. It's not one species that gave a lot of different species. So why does some genera do that and other not? And I take La Réunion because La Réunion and São Tomé are the two archipelago with the most number of orchids. If you compare that to Hawaii, there is only like 3 species of orchids in Hawaii, which is really small. And that's interesting because orchids have very long dispersal capacities. So why do they arrive in archipelago that are very close to the continental coast? And why not further away? That's another question that would be interesting to see. And of course, this work has been done with multiple actors, multiple persons. It was not just me in the field. So I want to thank all of them. And that's it.
Thank you very much, Tania, for this very detailed account of your research on the Tridactyl orchids. As a reminder, your jury members are Professor Barbara Gravendeel from Naturalis, Dr. Alain Vanderpoorten from the University of Liège, and Simon Delicourt from the Université Libre de Bruxelles. I would like to invite the jury members to come up with a series of questions, maybe 5 to 10 minutes each. And I would like first to invite Barbara Gravendeel to set the first series of questions. Thank you very much.
Hello. So, Tania, thank you very much for this very nice presentation. Now your orchids really came alive for me, and also the beautiful fieldwork that you conducted. So I was intrigued actually by your last slide, in which you mentioned that you would love to continue working with the Angiosperm 353 or even Orchidaceae 963 kit. So of course we all want to do work with these kits, because as discussed before, you can sample a larger part of the genome. However, these kits are not so suited to work with herbarium extracted DNA. And I think a lot of your collections, as we also discussed, are in musea. So how do you think you could tackle that particular challenge and not just use field collected orchids, which are becoming rarer and more endangered every day, but also employ the rich resources that we have in stock in our national musea?
Okay, so I think that if we can use the herbarium specimen to do like I did for the chloroplast and ribosomal data, and then compare the phylogenetic tree obtained with only the chloroplastic and ribosomal data with some obtained with nuclear markers to see if there is discordance or not. And then if there is discordance, then we can take with caution the chloroplastic and ribosomal data. But if there is no discordance, then we can just use the chloroplastic and ribosomal data set and use them with a lot of herbarium specimen included.
Okay, yeah, I agree. That would be a very good strategy. Actually, I think there's also an alternative, and that is that you could move beyond the 2 kits that you now mentioned, as they're new kits in the make with baits that are also optimal for annealing to ancient DNA from museum specimens. So I hope you're open to sort of an orchid kit 3.0, if you could mention it like that. That is the best of both worlds.
Yeah, well, definitely the research doesn't stop, and if we can find more markers that are more suitable for herbarium specimen, that's just great. Yeah.
I also mentioned this because, like you say yourself, you need more data to provide more evidence for your hypothesis of this back colonization, that these populations on Príncipe are ancient lineages that survived climate fluctuations and served as a sort of refugee area. So you really need to go to the population level. And do you think you can capture this fine-scale genetic variation with the current kits?
I hope so, but I'm not sure. I already had a lot of genetic diversity only with the 2 markers I used, so I think it's possible, but of course if you don't try, you cannot know. So yeah, but I'm pretty confident that it can be done for tridactyl orchids, yeah.
Based on your experience with the variation that you found in the chloroplast genomes?
Yeah.
So, so far we talked about genetic data. Can you also say a bit more about the morphology the morphological traits that you would like to investigate further if you would have the chance to continue working on this group of orchids?
Yeah, like the fruit, the pods. So. The pods of the orchids. When Tariq started the Shade House network, they focus on flowers because that was the most important at that time, having flower to identify the plants. But it's true that pods are also taxonomically important, and I think that now we have this idea of maybe for the species that we have a lot of individual in the different shade house, we can let some of them go into fruit and then observe the fruit, observe how they opened, how the fruit is posed, if it goes up or down or on the side, and do maybe physical test on how the pollen is dispersed based on the difference of the pod position. So yeah, a whole area of research is opening for the Shade House network. Yeah,
and don't forget the seeds inside the pods. So there's a very interesting publication that measured aerial dynamics of the seeds. And found strong differences between epiphytic and terrestrial orchids. So maybe these aerial dynamic differences are also present among the different species of your group, and they might explain their current distribution. I don't know, do I have more time, or is it polite to let others ask questions now?
You can have one more question.
One more question. Okay. Yeah, again, about your hypothesis of the back colonization, because you really need to know then what are the ancestral populations. So do you have any more suggestions for age estimates of the lineages that you now found in your molecular trees?
Well, concerning the Tridactyle-Cyrtorchis, no, I'm not sure. And there is also other question of maybe, but that's really a long shot guess, that there is a polyploidization between maybe, but again, that's a very long shot, between one species from West Africa, which is Tridactyle armeniaca, and maybe the population of Príncipe, because Tridactyle armeniaca is apricot in color, the population of Príncipe is white, and the population of Tridactyle-Cyrtorchis on the continent are white with the end of the petals orangey. But that's a very long shot, right? I have no clue at that stage, so yeah.
And have you tried crossing them, or have people tried crossing them in these greenhouse collection?
Not yet, but again, more work to be done.
Yeah. Thank you very much.
Thank you.
Thank you very much. Thank you very much, Barbara, for your questions. I'd like to invite Simon for his questions as well to Tania.
Oh, yes. Okay. Thank you very much, Tania. I really want to congratulate you again for this impressive thesis and for the amount of work that it represents, but also for having reached this important milestone. And today, I also really appreciated your presentation. I found it full of energy, so that was great to hear. As I had the opportunity to say during your private defense, I like the way you framed your thesis. I think that the resulting structure of your thesis benefits from this relevant connection between your different chapters. So this is there is a nice red line that you managed to follow during your thesis, and that's nice. There is a real storytelling between your chapters. Even if this is still a working paper, I was particularly interested by your chapter 3, as you can imagine, and I particularly liked the way you presented and analyzed the results from the APC analysis performed in that study. Again, I had the opportunity to say this during the private defense, but I think that the APC approach to visualize the outcomes of the simulations is a brilliant idea. In the context of this public defense, I still have 2 questions for you. So the first one is that while reading your thesis, I was actually curious about the motivations that pushed you to choose to work on these particular biological models. So if I rephrase my question, why working on African orchids in particular? And I precise that I wrote that question before the very nice introduction of your thesis that you did today. So yeah, I already have some pieces of answer to that question.
Okay, I can add that when I was in 2nd grade of biology, I said one day I will go to the tropical rainforest and find a new species of orchid. And then I find Tariq that was working on orchids. So it was like, oh wow, that's possible. It will be amazing. And so yeah, that's it.
Okay, and do you want to keep that biological models in the next coming years as a researcher, or would you also like to explore different biological models to explore evolutionary hypotheses or the research questions?
I really like orchids. I think they are really amazing, and their diversity is just incredible. And there is a lot of work to do on African orchids. So I would love to continue on orchids, but other plants really interest me. Maybe not more. I will say that like yeast, it's not really my big side in biology, but if it's plants, and especially orchids, I'm happy.
Okay, thanks. Yeah, and my second question, I mean, I already had the opportunity to discuss a little bit this aspect with you during the private defense, but in the context of the analysis performed in your chapter 3, you analyze 4 specific scenarios, which are different yet similar, as the 4 populations seems to systematically share a common time of divergence. Do you have some ideas of alternative phylogeographic scenarios that could be tested And is it part of your future perspectives to finish that chapter to include other scenarios to test?
Well, that would be great, but I don't know if we'll have the possibility with because it will imply to have more specimens, especially from Nigeria and Republic Democratic of Congo. Okay. And then actually what I've not presented is that we did a first analysis, just a RAXML analysis to see the different individual, and even if the tree was not resolved, we chose a different region based on that tree. It's the Cameroonian individuals were dispersed in the tree, and then the Gabonese individuals were all together, Príncipe one all together with some Cameroonian one, and the Guinean one all together. So that's why we choose the scenario that we have chosen. But yeah, if we have more specimens from different regions and more specimens from Gabon or from Guinea, the scenario can completely change from, and the AYABC do not allow migration after, so using like the AYABC ABC toolbox that allow migration would be interesting to see if it changed something. Probably it will. So yeah, it's a whole process to be still done.
So do you mean that after divergence, the do-it-yourself ABC program doesn't allow you to have gene copies exchange between the islands?
That's it.
Okay, so the only thing you could still do is to play with different time of divergence between the four islands, I guess.
Yeah, and well, there is other possibilities like merging two populations together to get a third one. So there is other slight possibilities, but okay. Yeah.
But I guess you don't see the point here of merging two islands.
But so maybe merging two regions could be done, like trying to see if we merge Guinea with Gabon, did we obtain Cameroon, by example? It's possible, but it's yeah.
Okay. Thank you very much for these very clear answers.
You're welcome.
Thank you very much, Simon, for your questions. I would like to know if the supervisor or co-supervisor, so Patrick Mardulyn or Tariq Stévart, have any question?
Alain is there. Alain is in the room.
Alain is in the room, but then he has another name, right?
Yeah, he comes from Liège for that.
Okay. Excellent. So Alain is with you?
Yeah.
Then Alain, we would like to know if you have any other question for Tania, please.
But first of all, it's very good to see your Tania, because it means that you managed to face all the problems that would have prevented you from achieving a successful PhD thesis. And I know that you succeed in cracking your university gates at night when they are closed. So in this thesis, yeah, the integrative taxonomy, so seeking for new characters in the genome, morphology, ecology of your studied species. But of course, you could also argue that the more you aggregate data, the more you will eventually find differences among taxa, but also population and individuals, and eventually all these could potentially lead to what is called taxonomic inflation. And thinking about orchids, this is certainly more true in that group than in any other group of land plants. And if you think of orchids, for instance, if you go to Greece, each Danish square kilometer has its own endemic orchid species named after some people in the local horticultural society. And so when do you think that we should stop naming things that do not look like their neighbors?
So I don't know if you heard the question of Alain, but it's basically with orchids, it's really different, and it's really difficult to know where to stop from varieties to species. And that's something I said during my thesis that Darwin already says on the birds in the Galapagos. So it's really difficult, and I think it's like, these days we don't see different subjects as binary, we see them more as spectrum. And I think from varieties to species, it's also a spectrum. And then the question would be, is that important for conservation of the species? Is that important to to address different questions. Like the species I described from São Tomé, they were clearly morphologically different from any others, and they were really well supported in the phylogenetic tree. When you look at, so I put the tree with one species per, one sample per species, but I did the tree with multiple samples per species, and they were not resolved to the point of one, to the to an extent that high. So I think that's a good way to see it, and then taking into account conservation purposes and question of the floral timing, the timing of flowering. That's another question that could be enter ecological questions too. So it's a whole range of questions that goes into the spectrum from varieties to species.
Well, as you know, in the past, taxonomy was actually the focus of a few people that basically spent the entire time looking at the specimen. Eventually, the question was much easier because they basically grouped specimens on the table depending on their genetic affinities, and they even didn't really think about all these questions about delimiting intraspecific taxa or species, or at which level they should decide that it's a given taxa or not. But increasingly now, taxonomy has become a more mechanistic process. I don't know if you have seen, but recently I saw there was a new journal, it's called Mega Taxa.
Oh.
The name is quite funny. And the journal promotes next generation taxonomy. And basically, if I summarize, they say that now the future of taxonomy is just to assemble an army of experts in their fields. First of all, NGS data, of course, because you need a bunch of molecular data to resolve your phylogenetic and cancer taxonomy. Then you need a bunch of other experts with skills in bioinformatic analysis. And finally, you need the third row of experts who are able to capture the pictures of specimens, do fancy morphometric analysis, and then use artificial neural networks to just identify a specimen like this. And as you know now today, with just a picture from your cell phone, you can give a name to anything living on Earth you can see. And so my last question for you was, what do you think of this next generation taxonomy? Is the old grandpa way of seeing taxonomy, as we saw during the past decades, completely over?
So the question is if next generation taxonomy is something a little bit made by old grandpa or not. Did I correct it?
I just said that before we basically grouped species based on their genetic affinities, and more and more it's more a mechanistic process that involves a series of experts in bioinformatics, production of genomic data, morphometric analysis, and use of artificial neural networks. I was just asking you, as a member of this new generation of taxonomies, what you think of this evolution of taxonomy?
Well, I don't think we need different experts, because I did the lab work for NGS, I did the bioinformatics for the phylogenies, and I did the taxonomical work. So some people are able to do the three together. So we don't need a bunch of experts in each field. It's true that it's through collaboration with other bioinformaticians, with other lab technicians, with other taxonomists that I arrived to this thesis, but it's through collaboration of different people in general, not different specifically involved scientists.
I mean, in this editorial I saw in the Gataxa, they basically argue that we are still very far from knowing the entire diversity of species that exist on Earth, and this is especially true in organisms like insects and lower plants. And so they say that if you want to save time, instead of accumulating decades after decades specific knowledge about a group, just hire a bunch of experts in the disciplines, so as I said, genomics, bioinformatics, image analysis, and then all together they will come out with a tool very quickly, with a tool that is so efficient that anybody with a cell phone could take a picture of anything and get a name after 10 seconds. So my question for you is, do you think that this is the right future for taxonomy?
Well, it works. I mean, there is this kind of application today on phone that do that, and I think we we can do that for a lot of species in Africa on orchids too. So yeah, that's maybe the future. But I really think that you need a little bit of knowledge on each subject to be able to be sure of what you're doing. I think you cannot just have one particular knowledge. Today it's more about maybe, of course, having a particular knowledge at a bigger level, but having some knowledge of the other disciplines to know what are their flows and what are their qualities. That's really important to do better work.
Yeah, of course. What we know is an idea of the uncertainty that we get. And when you take a picture of something and you ask a database, what's the name of this? You get a bunch of answers, but what is the reliability of this?
It's difficult, I think, for some flowers, for some plants it's possible with quite accurately, but like if you take the dandelions, I'm pretty sure they can't go to the species level, so it will probably depend on the species, on the type of organism that you look at.
So we still do need taxonomic expertise. That's what I wanted to hear. Thank you very much.
Thank you.
Thank you very much, Alain. And so I would like to ask the supervisor and co-supervisor, so Tariq and Patrick, if they have any other question before we proceed to our little meeting with the jury.
No, they have no questions.
They have no questions? They have...
The whole room can assure you that.
Okay. Do your supervisors have a way to join me for an online session with the other jury members now? And Alain as well, because Alain is obviously in the room there.
Yeah. They will go. You don't have internet here.
You can remind Nicolas that the public can ask.
Oh yeah, the public can ask question or two as well, Nicolas.
Absolutely, yes.
It's not me, it's Patrick that says it.
I know, I heard. So if anybody in the public has questions, that's the next step before going with the jury for a little meeting. Does anybody in the audience?
There is one question.
Yes.
It's all about radiation.
Uh-huh.
Is it radiation like in the common sense, or is it another?
So radiation is when one species speciate in different other species. So there is one common ancestor to multiple different species, like the bird in the Galapagos. They all came from one ancestor and they diversified between what they eat. So some with very big beaks eat very big seed, difficult to break, and some with very tiny beak are able to eat something else. And by diversifying what they eat, they diverge and they form new species from one ancestor. Yeah, that's it. Yeah.
Any other question?
No.
Now or never?
Yeah, Vincent de Blot.
Wanted to know what are your how as I would go half of the assembly. But if you had time to address pollination, do you think it will add some support or some that will help you to investigate your research question?
So the question is, if I have time to do pollinator study, would that help for the question that I address? Well, I don't think so. Maybe for two species from São Tomé that are sister species in the tree. And so one species is mostly found in primary forest and secondary forest, and the other one is found next to the sea. So because they are on the same island, maybe by finding which pollinator are pollinating them. If the pollinators are different, that might be a reason of diversification on the island. But otherwise, it will be more for other questions than the one I address in my thesis. Okay, I think it's good. Yeah.
That's it?
Yeah.
Okay,
yeah.
Good. Thank you very much, Tania, for your presentation and for the many answers to the many questions you've had today. The jury will group together now for a little chat. So I suggest you all stay there, and we basically leave this channel, and we will meet up on a separate parallel channel.
Patrick, Alain, and Tariq are going into the office of Patrick, so it will take them 5 minutes to arrive.
Okay.
There, just so you know.
Coffee break.
Yeah.
So stay there, and we will be back shortly. Okay.
Yeah, and he will call you back to the meeting.
Excellent. Thank you very much.
Thank you.
Have a good break. Thank you. Okay, so the jury has had a little group meeting, and we would like to congratulate you, Tania. I also personally thank the jury members, Barbara, Simon, and Alain, for their many questions and overall for the really positive evaluations. We all acknowledge the fact that you have made these complex concepts really super accessible for the general public. That's a really important step of your PhD thesis. You have conveyed most of your results with a really positive energy, so that's also something we really appreciated today. And overall, you have really embraced this challenge of a public defense really well, because that's a particular step where you really need to adapt your discourse and the way you present your results to a general audience. So based on those really super positive comments, and on behalf of the other jury members, I'm really pleased to tell you that your PhD thesis fulfills the requirements of your doctoral school, and you are therefore awarded the degree of Doctor in Biological Sciences from the University of Brussels, ULB. Congratulations, Tania. That's a really, really big moment in your life, and I would like to ask your supervisors, I think, have a few more words for you. So I would like to invite Tariq and Patrick. They may fight with one another at talking to you again, but so I would just invite them to say a few more words about their role as supervisors. Congratulations again, Tania.
Non. Oui, non, mais c'est pas en même temps. Je vais d'abord dire quelques petits mots, comme d'habitude. Et puis après, j'ai pris la peine d'aller dans mes archives du terrain. Nous verrons donc. Chère Tania, tout d'abord, permets-moi de te féliciter pour ta thèse qui est de grande qualité originelle et qui a été vraiment admirablement été présentée aujourd'hui. Bon, moi j'avais déjà vu avant un petit peu quand même. Tu as vraiment fait un super show et tous les membres du jury l'ont souligné. Okay, donc c'est vraiment très bien. Alors j'ai apprécié évidemment ton PowerPoint riche en couleurs, original, plein d'imagination. Et alors je me suis permis un truc un peu bizarre, mais je dirais un peu comme toi, si je peux me permettre. Ton PowerPoint te ressemble. Bon, on se connaît depuis ton master. J'ai vu une date là. Je suis pas sûr que c'est vrai, mais ça fait quand même assez longtemps. J'ai rien dit, mais voilà. Donc, et alors pendant toutes ces années, donc j'ai pu apprécier ça aussi. Ça a été souligné. Donc ta détermination, ta volonté, ton courage face à bon, j'ai de nouveau trouvé un mot diplomatique, aux nombreux changements qui t'ont accompagné, qui n'étaient pas toujours faciles. Hein, donc voilà. Mais te voilà docteur, hein, félicitations donc. Et c'est une grande étape de franchie. Je t'avais dit vraiment qu'il fallait aller jusque-là. Donc tu as d'autant plus de mérite que ton sujet n'était pas évident. Alors ça, je l'avais dit aux membres du jury, mais donc vous devez savoir que j'ai proposé à Tania de travailler sur les tridactyles. Parce que mon sujet de thèse à moi était plus ou moins le même que celui de Tania, mais j'ai abandonné parce que je n'avais pas de moyens techniques, c'était en fait trop dur. Et donc je me suis dit, qui peut y arriver? Et je t'ai proposé ce sujet, je suis très content, c'était un bon investissement puisqu'aujourd'hui on voit le résultat et donc tu as vraiment réussi à aller jusqu'au bout d'une tâche que j'aurais voulu faire moi-même. Alors, oui, et donc d'autant plus qu'il faut savoir que Tania, enfin vous la connaissez tous, elle est très indépendante et donc elle a vraiment réussi, elle a beaucoup de mérite parce qu'elle a vraiment fait beaucoup de choses par elle-même et le travail que vous avez vu aujourd'hui montre cette indépendance. Donc on l'a vu, hein, son travail a touché à de nombreux aspects. La morphométrie, donc elle a passé beaucoup de temps dans des herbiers à mesurer des trucs que personne ne voudrait mesurer. Puis après, elle a passé beaucoup de temps dans les laboratoires à faire des trucs bizarres que j'ai jamais compris. Et puis après, elle a passé du temps à faire des stats qui sont vraiment incompréhensibles. Et puis après ça, elle a quand même eu un peu quelques missions de terrain, dont une que j'ai fait avec elle. Et ça m'a permis d'aller chercher justement les photos que je vais montrer maintenant, qui illustrent bien ta personnalité. Voilà, elle a déjà peur. Et voilà. Donc il faut savoir, avant que je montre les photos, que Tania adore se prendre en photo elle-même, qu'en plus elle laisse toutes ses photos sur l'appareil photo du laboratoire. Et donc j'ai dans mes archives non seulement mes photos, mais aussi les siennes. Voilà. T'as vraiment peur. Hein.
Oui.
Mais non, j'ai été. Oui. Non, mais ça, c'est un petit dossier, c'est une petite partie. On va rester dans le. Hop.
Tu veux montrer aux gens de l'autre côté? Qu'est-ce que vous voyez ça comme... Ah oui, c'est ça. Voilà.
Ok, donc c'était en 2017. Là, on est au Gabon. Et donc, c'est une mission avec Joao. C'était dans les monts de cristal. Et donc, voilà. Donc là, elle est accompagnée de John. John, c'est le chauffeur du programme, mais aussi le type qui récolte, le type qui va en brousse et tout ça, et qui a accompagné Tania pendant 2 semaines, je pense. Voilà, ils se sont évidemment très bien entendus, donc vous le voyez. Et puis, à un moment, je ne sais pas ce que tu voulais lui faire, tu pourrais peut-être nous dire.
Mais... vraiment le pousser dans le
c'est ce que j'ai pensé. Voilà. Donc j'ai découvert cette photo que je n'avais jamais vue. Voilà. Donc ça, c'est les monts de cristal. Les monts de cristal, c'est l'endroit où il pleut le plus au Gabon. C'est tout le temps humide. C'est d'ailleurs supposé être un refuge forestier. Et voilà. Et donc, pour collecter des orchidées, vous aimez les voir avec les fleurs, pour les identifier. Et donc, c'est à la saison des pluies que c'est le meilleur moment. Donc là, on est en novembre dans les monts de cristal. Et comme vous pouvez constater, c'est rempli d'eau partout, il pleut non-stop et s'il pleut pas, là vous voyez les eaux, il pleuvait pas, mais les eaux qui partent. Et donc c'est pour vous montrer un peu les difficultés sur le terrain. Les photos sont inversées, ça n'a pas mis. on va la bouger, parce que je les avais bien remises, mais voilà. Donc, Tania vous a dit tout à l'heure que sa grande passion, ce qu'elle voulait depuis qu'elle était gosse, c'était de trouver une espèce et la décrire. Ben voilà, c'est cette espèce-là, la première qu'elle a décrite. Et donc voilà, donc vous voyez ce qu'elle voulait en faire, se prendre en photo avec son espèce. Voilà. Et là, ouais, franchement, vous la voyez de plus près. Donc cette espèce de truc qui ressemble à une boucle d'oreille bizarre, ben voilà, c'est ça qu'elle a étudié pendant quelques années, et c'est surtout cette espèce-là qu'elle a décrite, enfin la première. Voilà, alors là vous la voyez sur le terrain, donc vous pouvez remarquer qu'il y a un peu de boue en saison des pluies, et donc là on presse des spécimens. Donc elle a appris non seulement à récolter des orchidées, mais aussi d'autres plantes, et donc vous voyez ici c'est toute l'équipe avec EOAM qui est en train de récolter et de presser des spécimens. Là, vous la voyez dans un chablis, donc elle a montré quelques photos tout à l'heure, mais je voulais en remontrer. Donc ici, elle est occupée à photographier, vous ne voyez probablement pas, mais une de ces espèces. Donc ça vous montre un peu la nature du travail sur le terrain. Alors ça, c'est ce que ça ressemble après 3 semaines de récolte ou 2 semaines de récolte. C'est l'arrière de la voiture et tous les sacs que vous voyez là sont bourrés d'orchidées. Et donc on les garde dans des sacs comme ça qui sont aérés, comme ça les espèces, les individus ne s'abîment pas. Donc ce sont toutes des orchidées vivantes. Et donc voilà, ça c'est les récoltes de botanistes après 1 ou 2 semaines de travail. Voilà, donc ça c'est... Ah, j'ai mis cette photo, je n'y avais pas pensé. Donc Tania a hésité entre continuer la recherche ou devenir grumier, pilote de grumier. Et donc voilà, et je pense qu'elle a continué la recherche. Bon, on l'a quand même accompagnée, nous aussi on a hésité à un moment. Mais donc là on était dans une concession forestière et vous voyez, ils sont en train de faire le débardage des arbres et on a profité de l'occasion pour faire quelques photos de groupe. Voilà, une autre chose, donc ça c'est un fruit de je sais pas quel arbre, et donc vous voyez que Tania est fort intéressée. Voilà, ça c'est le truc, le petit pangolin, donc qui est devenu célèbre puisque à cause du Covid, tout le monde en a parlé à un moment, et grâce au Covid, ces animaux qui sont en voie de disparition le sont un peu moins, donc c'est un des effets positifs du Covid. Alors, encore une photo avec pour les gens qui connaissent un amorphophallus, donc Tania qui trouve que cette plante extraordinaire est je ne sais pas quoi, il faudra un jour que tu nous expliques, on n'a pas compris, n'est-ce pas? Donc après le Gabon, on est passé à São Tomé, donc vous en avez beaucoup entendu parler aujourd'hui, donc ça c'est un paysage au nord de São Tomé. Voilà un autre endroit, donc São Tomé c'est un endroit vraiment difficile à vivre, comme vous pouvez le constater. C'est vraiment des conditions dures, surtout au nord, où il fait toujours beau. Et voilà, non, en réalité c'est une île volcanique, donc le travail sur le terrain est très difficile. Mais le gros avantage, c'est que, voilà, ça c'est au nord de São Tomé, il y a toujours du soleil ou presque toujours du soleil, et donc quand on est complètement déprimé et fatigué, c'est là qu'il faut aller, et ça permet de reprendre des forces. Voilà, donc avec Joao, encore avec Joao devant un petit arbre au nord. C'était après une mission de terrain un peu compliquée, difficile. On s'est dit, maintenant on va faire du tourisme. On en a profité, on s'est promené. Et puis ça, c'est sur le terrain. On fait aussi du tourisme, mais bon, un peu moins. Voilà, donc São Tomé, c'est un volcan, donc il y a tout le temps des rivières, des cascades et tout, donc c'est quand même assez sympa comme endroit, mais les genoux souffrent. Alors voilà, ça c'est le repas. Non, alors il y a 2 photos liées à ce repas. Il y en a une de moi qui est franchement ridicule, et puis il y en a une de Tania que je vous laisse apprécier. J'ai oublié de mettre la mienne. Mais je la connais, celle-là. J'ai eu le plaisir de la revoir. Voilà, tout à fait. Alors ça c'est la nuit, donc elle a dit qu'elle ne s'était pas intéressée aux pollinisateurs, mais on était quand même intéressés parce que comme personne ne sait qui pollinise les tridactyles, on était là avec Joao, on aurait bien voulu savoir. Bon, c'est grâce à ça qu'il y a eu quand même quelques photos qui ont donné des pistes, mais c'était pas évidemment le but de son étude. Cela dit, se trouver le soir comme ça, bon là c'est dans un jardin botanique, mais c'est quand même très sympa et ça permet de faire des observations. Alors ça, c'est Tania dans l'appartement qu'ils ont occupé pendant un mois et qui découvre qu'elle n'est pas la seule à avoir étudié la spéciation dans les îles du Golfe de Guinée. Bon, il y avait quelqu'un qui avait étudié les oiseaux, donc elle était très contente de savoir qu'il y avait déjà des hypothèses et d'autres. Et voilà, ça c'est pour finir. Donc vous voyez, elle fait son show comme à chaque fois. Et donc voilà, donc ça c'était quelques photos. Ce qui est très agréable avec toi, c'est qu'il y a plein de photos et donc quand on doit sélectionner, j'ai vraiment eu l'embarras du choix. En tout cas voilà, je te félicite encore pour ta thèse, pour ta présentation et j'espère que ces quelques photos ont permis de voir qu'est-ce qui se passait en Afrique quand tu n'étais pas avec tous ces gens, quoi. Bon.
Moi, je propose qu'on commence par te faire signer ton diplôme. Normalement, c'est le président du jury qui fait ça, mais comme il est à distance, donc, je te propose de signer ici. J'en ai un si tu veux, parce que ce serait mieux de ne pas utiliser un bic rouge. Ah.
Oh, wow.
Bon, Tania, voilà, ça y est, on y est, malgré les doutes, malgré les craintes, le stress de dernière minute, ça y est, tu es docteur en sciences aujourd'hui. Toutes mes félicitations. Je pense que tu peux être très fier de ce que tu as fait globalement. Je pense que tu peux être très fier de ta thèse. Finalement, tu as remis une thèse qui mélange harmonieusement la taxonomie, la phylogénie, la biogéographie et même la phylogéographie. Donc je pense que ça forme vraiment un tout très, très chouette, très intéressant, très diversifié. Évidemment, ce parcours n'a pas été un long fleuve tranquille. Hein. On va peut-être pas revenir sur les étapes, les obstacles compliqués. Ça a été un petit peu compliqué au début. On va pas revenir là-dessus, mais je pense qu'on peut peut-être se revenir par contre sur les points positifs. Après un an, tu as présenté le FRIA et tu as réussi à obtenir ce FRIA, à obtenir une bourse. C'est pas donné à tout le monde. Ensuite, je pense qu'il y a eu Tariq en a déjà parlé, il y a eu beaucoup de travail au laboratoire, de travail dans l'herbarium. Tu as eu l'occasion de partir dans des très beaux endroits, à des congrès aussi, je crois que tu as participé à beaucoup de conférences. Je pense que tout ça c'est positif. Essayons de retenir ce qui est positif. Je pense que tu as beaucoup appris ici. Alors, évidemment, ça a terminé avec ce foutu Covid qui t'a un petit peu chamboulé. Ça a été, je pense, très dur pour toi, comme pour d'autres personnes également. Et ça ne t'a pas aidé pour terminer, mais tu y es arrivé. Donc, je pense que c'est quand même ça l'essentiel. Pour ma part, j'ai commencé à interagir avec toi plutôt dans la deuxième partie de la thèse. Donc, j'ai pas eu la chance de partir sur ce terrain. J'ai pas de belles photos à vous présenter, comme Tariq a pu le faire. Et c'est à ce moment-là que j'ai appris vraiment à te connaître. Si je dois trouver une caractéristique qui te convient très bien, et en fait sans m'être concerté avec Tariq, j'ai entendu le même mot qu'il a utilisé, c'est que tu es quelqu'un de déterminé. Ça, pour moi, c'est vraiment quelque chose qui m'a marqué. C'est que quand tu veux quelque chose, tu fais tout pour l'avoir. Et ça, c'est vraiment très bien. Une des premières choses, une des premiers comment dire, un des premiers événements qui m'a fait que je m'en suis rendu compte, c'est que quand tu es venu chez moi au début, tu t'es dit, tu voulais utiliser des caractères moléculaires pour faire de la phylogénie, de la phylogéographie, de la biogéographie. Et d'abord, tu as commencé par te focaliser sur des marqueurs chloroplastiques. Et moi, j'ai d'abord essayé de te convaincre en disant, mais ce serait quand même mieux d'avoir plusieurs locus indépendants, le locus nucléaire. Je pense que je t'ai plus ou moins convaincu à ce niveau-là. Tu n'as pas été difficile à convaincre. Mais par contre, je t'avais aussi dit, la meilleure façon de faire, à mon avis, c'est d'utiliser une approche classique d'utiliser des amorces en comparant des séquences d'autres gènes qui existent déjà et puis en essayant de les amplifier chez tes espèces. Bon, tu as essayé de faire ça pendant quelques temps. Puis tu as fait comprendre que c'était bien gentil, mais que c'était quand même un peu ringard, qu'il existait aujourd'hui des techniques de nouvelle génération de séquençage qui étaient vachement plus modernes et que tu avais vraiment envie de faire ça. Et tu as été vraiment déterminé et tu as trouvé le moyen, tu as trouvé les bonnes personnes pour le faire. Grâce à Olivier et son équipe, tu as pu te lancer là-dedans. Et je pense que ça a été payant, puisqu'effectivement, ça a bien pu faire avancer ta thèse, et tu as pu aujourd'hui produire des données qui vont pouvoir être publiées et qui ont donné des choses vraiment très intéressantes. Et je pense que cet épisode, ça reflète un petit peu une de tes grandes qualités, c'est que tu es capable d'aller toujours trouver les personnes dont tu as besoin pour faire avancer ton projet. Ça, c'est quelque chose pour moi qui ressort de ta thèse, c'est que tu vas chaque fois trouver les bonnes personnes pour faire avancer ton projet, quand tu reconnais volontiers que tu n'es pas experte en tout, et tu vas trouver les experts qu'il faut pour pouvoir progresser. Et tu as fait ça tout au long de ta thèse, et ça t'a bien réussi. J'ai quand même un petit copion ici. Alors, globalement, j'ai aussi appris que tu étais quelqu'un d'extrêmement sociable. Je crois qu'ici, on en a la preuve. Et il y a encore plein de gens qui se sont connectés, et d'autres qui ont essayé, mais qui ne sont peut-être pas parvenus. Mais bon, ça, c'est vraiment la preuve. Tu es vraiment quelqu'un tu as besoin de contact humain. Alors, c'est vrai sans doute pour un peu tout le monde, mais il y en a certains pour qui c'est beaucoup plus que d'autres. Et je pense que Tania, c'est vraiment quelque chose. Elle ne peut absolument pas se passer. C'est probablement une des raisons pour laquelle la crise du Covid a été... je ne dis pas que c'est la seule, mais c'est une des raisons pour laquelle cette crise a été si difficile pour toi. Mais c'est une bonne chose. Alors, je t'ai trouvé quelqu'un de très sociable, comme j'ai expliqué tout à l'heure, capable d'aller trouver les bonnes personnes quand tu avais besoin d'expertise que tu ne possédais pas, mais j'ai aussi vu que tu étais quelqu'un de très généreux. Tu es très sociable et à la fois très généreux. J'ai vu, il y a juste 2 exemples, celui de Maïva que tu as aidé pour venir s'installer ici, celui d'Estelle que tu as accueilli pendant son stage ici. Donc voilà, ça me montre que tu es quelqu'un de très sociable et de très généreux. Donc voilà, l'histoire ne s'arrête pas là, parce qu'on a encore un peu de travail à faire ensemble. On a encore au moins 2 articles à publier. Il y en a un qui est en révision. Je pense qu'on sait ce qu'il faut faire pour parvenir à le faire accepter. On y arrivera sans problème. Le deuxième sur la phylogéographie, bon là il y a peut-être encore un petit peu de travail, mais j'espère si tu en as envie qu'on pourra tous ensemble réussir à le faire publier aussi. Donc on va encore se voir, mais néanmoins j'espère que tu vas vite. Je pense que ton souhait maintenant c'est de trouver un post-doctorat pour pouvoir continuer ta carrière scientifique, et je te souhaite vraiment de trouver très rapidement un poste. Tu le mérites absolument. Voilà. Encore toutes mes félicitations. Oui, pardon. Je vais te rendre le micro.
Merci beaucoup. Oui, merci à tous les deux de m'avoir accueilli, Tariq un peu plus tôt que Patrick. Je me rappelle encore de Patrick qui était dans mon jury de mémoire de master et qui était incapable de prononcer le nom tridactyle, et j'espère qu'au moins ces 6 ans lui permettront de connaître les tridactyles un peu mieux. Oui, merci du fond du cœur de m'avoir accueilli, parce que Effectivement, c'était un rêve, peut-être pas d'enfant, mais en tout cas de bachelier. Ceux qui étaient avec moi en bachelier s'en rappellent de cette fois où je me suis retournée en disant « Ah, nouvelles espèces d'orchidées dans la forêt tropicale » et où ça m'est arrivé 2 ans plus tard avec le master qui me permettait d'aller au Cameroun, etc. Et puis Patrick qui m'a récupéré au vol, littéralement quand j'avais besoin d'un promoteur qui s'y connaissait en génétique. Et donc voilà, et sans vous, j'y serais pas arrivée. Donc ouais. Merci du fond du cœur. Et bon, unfortunately for the ones that are not in Belgium now, you're not gonna be able to celebrate with us, but pour ceux qui sont en Belgique et qui éventuellement veulent venir depuis Internet boire un verre au Square G, c'est possible, et pour les autres dans la salle, c'est définitivement possible.